Hey guys, welcome back to the channel. I wanted to talk about a couple of different ways in which we can measure the brain electrical activities. And in this channel, we have primarily talked about EEG, but there's another way that you could also measure them called LFP, or local field potentials. And there are some differences, some major differences, both in terms of how you measure them, but also where in the field of science you use these measurements. What are the pros of one in comparison to the other, as well as what are the cons of them. So hopefully by the end of this video, you will be able to learn the difference between the way in which we measure the brain's electrical activities. Okay, so let's start off with EEG. Although we have talked a lot about EEG in this channel, let me just refresh the memory. So when it comes to EEG, essentially it's extremely non-invasive. In other words, you don't have to poke into the brain to put the electrodes. You can put sensors on the scalp of the brain, and in that you can measure whatever electrical activities that comes out of the brain from that you can measure from the scalp of the brain. You can actually measure it that way, and that is what we call a non-invasive procedure, because again, you don't have to poke into the brain. And the traditional ones, you have 30 plus number of sensors, but then there are also commercial neuro headsets. The one that I use primarily called Emotive, which has 14 sensors. Now let's talk about LFPs. In LFP, you also measure brain's electrical activities. The difference is that you insert electrodes in the brain tissue of the subject. Mostly it is done on animals. I think primarily it's done on animals, but the big difference is that it's invasive. So you have to operate on the subject, on the animal, to insert it into the brain. I've never done LFP myself for various different reasons that we don't need to go into, but the way in which, again, it's measured is from the electrodes that are inserted. So it is needless to say that when you use LFPs, you can really penetrate into the brain tissue and get much more accurate local brainwaves activities. Let's now look at the difference in terms of the type of data that you can collect from EEG versus LFP. So EEG, as we said, it's non-invasive. So you can gather the brain electrical activities from the scalp of the brain. So let's say, for example, you can say a prefrontal lobe exhibited mainly beta waves, for example, you can say that. Whereas with LFP, because you have these sensors implanted within the brain tissue, you are able to localize the type of brain activities that you're collecting much more accurately than you would have with an EEG device, because you are inserting the electrodes within that local part of the brain, a specific part that you're interested in. The pros and cons with EEG versus LFP is that when it comes to EEG is much faster. We call it a better temporal resolution, because as soon as you get brain electrical activities, these sensors can pick it up, and they can pick it up from a large region of the brain, because again, it's from the scalp. Whereas LFP, they say that it has a better spatial resolution, meaning to say that it is able to more accurately say which space of the brain where these electrical activities were exhibited. So that is one of the bigger differences in when you compare EEG versus this is one of the reasons why you see in the field of sciences the way the EEG data is used versus LFP data is used differs. When it comes to EEG data, mostly it is used in the health related brain disorder related type of analysis. For example, you can detect epilepsy, seizures, other type of brain disorders, etc., with EEG. Because it's much faster, you'll be able to pick up the signals much faster, and also because you don't really need to, you only need to determine the excitement of a bunch of neurons to be able to detect, for example, seizures. On the other hand, LFP is much more used in the type of research studies that scientists do that has to do with behavioral analysis, neuroscience analysis, decision making, etc. All those kind of areas, and that's because you're able to, once you do your experiment and also once you write your paper, you'll be able to distinguish exactly where in the brain that you were collecting the brain activities from, and that becomes much more sort of, relevant when you are studying behavioral analysis. And that's what you do primarily, for example, animals, and then you draw inferences onto humans. Okay, guys, I hope that you found this video helpful. If you did, don't forget to smash that like button and also the subscription button. I would really appreciate it, and I'll see you next time. And Merry Christmas, by the way.